Thank you very much for having me here today. I obviously have not coordinated with Anna, so you'll see some repeats of what Anna just said. So let me start with the most important slide, is the team that actually is behind a lot of the work I'm going to talk about. But main person working is Andrea, who is going to give a talk a little bit later. I also want to acknowledge our funding sources. Let me start with the problem. I know we all hear about CT and NB1, we're all very familiar with it, but the problem is actually bigger than that. The genetic disease, and many people don't appreciate it, affect more people than HIV and cancer combined. There's over 7,000 diseases. Every year, over 8 million children are born and that are affected by the disease. And unfortunately, about 30% of them will not live to celebrate their 5th birthday. So it's a massive global problem that people simply don't talk about enough. But there's some exciting times. So there's a good news that I want to share with you as well. Many of us have actually witnessed 3 very important revolutions. The first one is recombinant DNA revolution, where we can actually take DNA, cut it, and kind of glue it together. So we can move genes, we can move things from one species to another, we can modify things, create things. That was a big change in the field. The second one, more recent, is DNA RNA sequencing revolution, where we can actually take the patient and very rapidly read the genome and try to understand what genetic changes may be responsible for the disease. And that's very important for first step towards the actually being able to find treatment. And more recent is genome engineering. Again, you're going to hear about it, some of that later, less during my talk, but this is our ability to now go into the cells, target the mutations, and correct them right in the genome. But the question is, with all this progress, what is actually the impact on genomics and the medicine? So I like this very simple graph where I show that our knowledge has progressed, is growing exponentially over time. Our ability to diagnose patients is following a very similar path. But unfortunately, our ability to use this knowledge to prevent or treat disease is not there yet. There's this gap that has formed, and that's what I believe is where gene therapy has an opportunity to bridge that gap. And in this context, the gene therapy is, the simplest definition is the use of genes as medicines. So this is a very simplified version, just I want to make sure that everyone has a basic understanding of biology here. Everything about us is encoded in our DNA, in our genes. A healthy gene produces a healthy protein, which then has a function and a normal expected function. A mutated, broken gene makes a broken protein and leads to a disease. Now, human genome is composed of 4 letters, A, T, C, and G. It's a very long sequence. It's about 1,000 times the length of a Bible or War and Peace. So it's a really, really long stretch of information. A single mistake, a typo, a change of letter, insertion or deletion of the letter could mean a difference between healthy and an affected sick child. Anna touched on this. There's different strategies when it comes to gene therapy. This is severely oversimplified. But gene replacement or gene addition, those are actually not really precise terms. The proper term is gene augmentation. This is where we provide an extra copy of the gene. This extra copy that we deliver into the nucleus of the cell never makes it into the genome. It actually just kind of, in our strategies, hangs around and is able to, the cell can use the information that we provide to produce the protein that actually has the function. We can also go into the genes and do genome editing. This will be affecting the patient at the mutation site. You also heard about ASOs. This is where we can actually modify, so from DNA, you make RNA. ASOs work at the RNA level. They can either knock it down or they can increase the expression. And the last type of therapy is protein replacement. This does not fall into the gene therapy umbrella, but it is also something that we apply in some of the diseases where we actually provide the protein that's missing. In the context of CT and NB1 and my lab, we are mostly concentrated on gene augmentation and gene editing. So again, gene replacement, as I mentioned, it is delivering of a copy of the gene, in this case CT and NB1, to the patients. There's many ways to deliver the copy to the target cells, and it's a critical, as Anna mentioned, that this has to be done effectively and very specifically, so it's safety and efficacy. For this, we use viruses. So we use viruses that we can then use to package the information and deliver it to the human. In simplified terms, You can imagine that you have a medication and it needs to be delivered to someone who has a headache, somewhere in this apartment. All we need now is an Amazon with a correct address. We package that into the box, and this travels to the correct apartment. This is, in very simple terms, what we're trying to achieve in the lab and in the clinic. This brings me to the term that I like to use is molecular medicine. Imagine somebody with a lung disease. The typical approach currently would be use as surgical tools, some surgeon, and potentially a new lung. So we will have to replace the organ. But now we have other options. We can use genetic tools, vectors, and new genes to correct the disease. So this is a new wave of medicine, and this is what we're trying to do for CT and NB1. I know it sounds a little bit sci-fi. Anna already kind of spoiled it for me, but there are examples right now that are worth mentioning here. The first one, Zolgensma. I'll come back to that one later, so kind of pay attention to this one. The second one is Luxturna. Zolgensma is a treatment for spinal muscular atrophy. It's a vector that encodes the correct copy of the gene that is given to the patient, travels to the brain, and expresses the protein that's missing in the child, and leads to devastating results. The second one is Luxturna, very similar approach, but for a genetic, for blindness with a genetic cause. What's important here that in both cases is the AAV vector that's the main carrier of the information to the target cells. And that brings me to AAV, which stands for adeno-associated virus, or as we like to call it, almost a virus. It is almost a virus because this is the little guy here. It's not this big, powerful guy here. It's that little guy that stays in the shadows. It is almost a virus because it actually needs to, in nature, it cannot work on itself. It has to, it depends on co-infection with another virus. So it depends on another one. It's called a dependovirus. Has a quite simple genome, and I'm not going to talk too much about it, but there is part of the genome that controls how the vector behaves, and the second part here controls the structure of the shell, the outside of the virus itself. And it is this shell that determines this little perking things on the side that determine which cell the vector is going to go to, and where it's going to deliver the therapeutic cargo. This is what my lab actually specializes in, is making new version of this to make them more efficient at delivering therapeutic payload to the correct cells. Now, when we take the virus, Anna already mentioned AAV is not pathogenic. About 70% of people have been infected with AAV in our lifetime. Nobody has ever been sick. So it's already safe. Nonetheless, we take the genetic material from the virus and we remove it. So we put it on the side, and that makes the space for our therapeutic cassette. That gives us about 4.4 kb of space. I know this number may not mean much to you, but it's important because you heard yesterday that CT and NB1 is actually quite a big gene. It's about 23 kb in length. So that's about 5 times bigger. That means we cannot fit the gene itself into the vector. However, genes are composed of 2 elements. The green boxes represent what we call exons. This is the important information. The things in between are spacers. They're actually, in simple terms, they don't have any function in this context. If you think about this being a movie, those will be the commercials. We can cut them out. We don't have to watch them. So we can actually take the gene and collapse and only use the important information. The genes don't exist in isolation. In a genome, you have other elements. Some elements that actually have a positive function on the gene. So this will be the engine that actually drives expression of the gene. But there will be also things that we call enhancers, as well as inhibitors. Those are the things that control how much of the gene is being expressed and where and when. So this is the where in correct cells, when, at which stage, and how much do you want to produce. When we try to move this very complex system into the vector, that becomes a little bit difficult if we don't have enough information. But in principle, we try to take the positive elements and put them in a vector. Obviously, we have to put the gene, and this just terminates the production. So this is just the ending. Then we have to package that in a vector. You can think of the vector as a casing of a pill that determines where it's going to go. Is it going to go to your stomach, to your intestines, or to your brain? And then obviously we want to make sure that those things get delivered to the target organ of choice. So I'm going to bring back Zolgensma again. Zolgensma uses AAV9. So this is AAV serotype 9. There's many different serotypes. 9 is particular because when given systemically into the bloodstream, it can actually travel, cross the blood-brain barrier, and enter the brain, which is where we want our therapy for CT and NB1 to go. This is what Zolgensma, we knew this in animals. Zolgensma was the first time to prove it that this actually works in humans. So this is groundbreaking information, and we're going to use it in our program. So what we're going to do is we're going to take what Zolgensma has proven, and we're going to use it, or reuse it, a lot of it, to create a therapy for CTNNB1. Again, Zolgensma has the vector, very similar to what we have, puts it into the patient, and it travels into the brain, opens up, brings the genetic information that mimics what is in genome. Again, we said the little boxes are gone. It pretty much looks like this, but it mimics what the nature created and produces functional protein. So from the physiological, from the cell perspective, the cell cannot tell the difference between the endogenous gene and the one that we delivered. It makes the same functional protein. So what our program is, and you're going to hear about it a little bit later from Andrea's talk, is actually reuse what Zolgensma does, pretty much remove the SMN1 gene, which is the gene that's affected in SMA, and replace it with CTNNB1. Now, the combination of all the elements that I mentioned, the enhancers, promoters, are a little bit tougher more difficult to predict. So we actually created 6 different cassettes, six different constructs that we tested to select the best candidate. Again, this is, I don't want to spoil it for you. Andrea is going to talk about it in a little bit. What's also important to understand that we scientists can do what we do, and we're pretty good at it, but we cannot work in isolation. We work very closely with clinicians and families to understand what actually is needed. And only when we understand the disease, which is not our expertise, and actually what the families observe and what the children will benefit from, we use this information to create the vector that has a higher chance to benefit the patients in clinic. But why are we doing this? This is a lot of work, and we're doing this for this. So we work with other patients. Some of you know Urban's pictures are here. So we work with CTNNB1 patients, but we also work with patients affected by other mutations. To be able to help them, we created what we call Advanced Therapeutic Translational Program, ATTP. This is a set of critical expertise all within 3 buildings from each other. So within 10, 15-minute walk, where we have research facilities, such as my laboratory. We have a clinical manufacturing facility. So once we have a candidate, we can actually produce it in the clinical grade. And then we have a clinical facilities. We also have a dedicated scientific advisory committee to the Human Ethics Committee. This is the committee that can actually approve the therapies. So we don't even have to go to the government. This all can be done at the hospital level. That means that we can now take a patient all the way from detection all the way to correction with a dedicated team of experts and specialists on our campus. Now, everything sounds quite simple, but it is quite complicated. There's a lot of expertise that has to come together. This is just a simplified snapshot of my lab, the different expertise that we had to build to be able to do this. We have programs developing new vectors, again, for different organs that we want to deliver the therapeutic to. We work on ASOs. We work on disease models. When you work with a disease that's not very known, you need to very often create the model, like such an organoid or transgenic animal, on which you can test and validate your therapy. We do a lot of bioinformatics and machine learning, genome engineering. We look for new viruses, new vectors that can replace AAV to make the therapy more efficient, and we also are very involved with manufacturing. This is also something that you cannot build within one country. We recruit expertise from all over the world. This is the current snapshot of nationalities that are in my team that come together to do this work. We also use the latest technology. I don't know if this is going to play, yes. So we use the latest technology to validate and increase the safety of therapy. So this is a mouse brain, and you can see it's a 3D reconstruction. All the green cells are the cells to which the vector delivered a marker, is GFP, so it's green protein. So we can actually test, before we go into patient, where the vector is going to go. Is it more efficient or not? We're also looking at manufacturing. All the different genes that we package, they package differently. So we look at how our therapeutic cassette would enter the vector, how it's going to look inside. So this is a reconstruction of the 3D, how it looks inside of the virus before it goes into patient. And that's very important for packaging, to make sure that the manufacturing is not very expensive, that the patient can afford it, but also for safety and stability, because of stability of the vector. This is just, I think this is the last slide. This is the slide that outlines the project that we are working on. When we started working with CTNNB1 Foundation, Spela and Anna, this was a new disease to us. We did extensive literature review to understand the disease, to understand what's known about it. We then created a number of constructs. There's 6 of them, and you'll hear about that later. based on all this information and our internal understanding. So yeah, so those different regulatory elements come together. We studied how this affects our ability to produce the vectors in clinic, and then we perform functional validation in organoids and 2D neurons derived from Urban, so from Spella's son, so from CTNNV1 patients. We are now moving into large-scale manufacturing in support of toxicology studies and animal toxicology study, both in-house as well as outsourced to an expert organization that specializes in that. And with that, again, I want to thank the team that works on it. But I want to leave you also with this. I really like this quote, and it resonates with me very well. There's a very famous saying that it takes a village to raise a child, but I really think it takes an army to cure one. And it's people like yourselves and everyone out there that has to come together in order to make this a reality. Thank you very much.